Memory is one of the most remarkable and enigmatic functions of the human brain. It shapes who we are, colors our experiences, and influences nearly every decision we make. From recalling your first childhood adventure to remembering complex equations, memory is the invisible thread weaving together the fabric of your life. But unlike a computer that stores data in fixed files, the human brain handles memory in a far more dynamic, intricate, and sometimes puzzling way. At its core, memory is the brain's ability to encode, store, and retrieve information. Encoding transforms experiences into a language the brain can understand. Storage preserves them over time, while retrieval allows you to access them when needed. Yet these processes are rarely straightforward. Memories are constantly evolving. They can strengthen, weaken, or even vanish entirely. This fluidity is why you might vividly recall the details of an emotional event from years ago, while forgetting mundane facts almost immediately. The human brain is astonishingly selective about what it remembers. Attention acts as a gatekeeper, determining which experiences are significant enough to be encoded. Emotions play a powerful role, often amplifying memories that carry joy, fear, or sadness. Chemical messengers like adrenaline and cortisol flood the brain during emotionally charged events, imprinting those moments more deeply than neutral ones. But memory is not just about storage, it is also about adaptation. Forgetting is not a flaw, it is a crucial feature. The brain continuously filters out unnecessary or outdated information to prevent overload, making space for new experiences and learning. In a way, forgetting is as vital as remembering, allowing us to focus on what truly matters. Despite centuries of research, memory remains one of neuroscience's most fascinating mysteries. Scientists continue to uncover how neurons communicate through synapses, how brain structures like the hippocampus and amygdala encode experiences, and how sleep and molecular processes consolidate or erase memories. Each discovery reveals not only the power of memory but also its delicate fragility. Understanding memory is more than a scientific pursuit. It is a journey into the essence of what makes us human. Every memory, whether fleeting or enduring, shapes our identity, influences our choices, and defines the way we experience the world. In this exploration of the brain's incredible capacity, we begin to grasp the mystery of memory, a living, evolving archive of everything we are and everything we have been. Every memory begins with a spark, a single moment, a fleeting experience, a thought that the brain decides is worth remembering. This initial step, known as encoding, is the process by which your brain transforms experiences into a form that can be stored and later recalled. Encoding is far from passive. It is an active, complex process that involves multiple brain regions, networks of neurons, and chemical signals. At the most basic level, memory encoding begins with neurons, the fundamental building blocks of the brain. Neurons communicate through tiny gaps called synapses, sending electrical and chemical signals that represent information. When you experience something, a breathtaking sunset, the taste of chocolate, or a profound idea, thousands of neurons fire in coordinated patterns. These patterns form the foundation of a memory. The more neurons involved and the stronger their connections, the more robust the memory is likely to be. One of the key processes in encoding is synaptic plasticity, the brain's ability to strengthen or weaken connections between neurons based on activity. When neurons fire together repeatedly, their connection strengthens, a phenomenon known as Hebbian learning, often summarized as neurons that fire together wire together. This strengthening is essential for forming lasting memories, turning fleeting experiences into retrievable information. Attention plays a critical role in determining which experiences are encoded. The human brain is bombarded with sensory information every second, but it cannot store everything. Focus and attention act as a filter, selecting which stimuli are meaningful enough to encode. For example, you might vividly remember a friend's wedding but forget the color of the car you passed on the way there. Attention ensures that what is important or emotionally significant gets prioritized for memory storage. Emotion is another powerful amplifier of memory encoding. The amygdala, the brain's emotional center, works in tandem with the hippocampus, which is crucial for forming new memories. Highly emotional experiences, whether joyful or traumatic, trigger the release of chemicals like adrenaline and cortisol, which enhance the brain's ability to encode these moments. This is why you may clearly recall your first kiss or a frightening accident years later, while more mundane events fade into obscurity. Memory is also categorized into different types during encoding. Sensory memory captures fleeting impressions of the world for a few seconds, like the sound of a car horn or the flash of a passing bird. Short-term memory holds information temporarily, typically for a few seconds to minutes, allowing you to remember a phone number long enough to dial it. Long-term memory is where encoding truly becomes transformative, as the brain works to consolidate and stabilize information for months, years, or even a lifetime. Interestingly, memory encoding is not always perfect. The brain is prone to errors and distortions, often influenced by prior knowledge, expectations, or context. This malleability is why memories can shift over time or why two people can remember the same event differently. The birth of a memory is an intricate dance between neurons, synapses, attention, and emotion. From the first spark of experience to the strengthening of neural connections, encoding transforms fleeting moments into the foundation of who we are. Understanding how memories are encoded not only illuminates the remarkable power of the human brain, but also opens the door to improving learning, emotional intelligence, and self-awareness. Memory is not stored in a single location within the brain. Instead, it is distributed across multiple regions, each specialized for different types of information. From remembering facts and events to recalling emotions and skills, the brain orchestrates a complex network that ensures our experiences are encoded, consolidated, and retrievable when needed. Understanding these brain structures provides a window into how memory works and why it can sometimes fail. At the center of memory formation is the hippocampus, often described as the brain's librarian. Located deep within the temporal lobe, the hippocampus is essential for forming new memories, especially episodic memories, which are personal experiences situated in time and place. Without a functional hippocampus, a person can experience anterograde amnesia, the inability to form new memories, although past long-term memories may remain intact. The hippocampus acts as a temporary storage site, organizing information before it is sent for long-term storage in the cortex. The cerebral cortex plays a key role in storing long-term memories, particularly semantic memories, which are facts, concepts, and general knowledge. Unlike episodic memories, which are tied to personal experience, semantic memories are distributed throughout the cortex in a networked manner. For example, your understanding of a city's landmarks, historical events, or the rules of a game is stored across multiple cortical areas, integrating sensory, spatial, and conceptual information. This distribution makes long-term memory highly resilient but also vulnerable to partial loss if certain cortical regions are damaged. Emotionally charged memories rely heavily on the amygdala, a small almond-shaped structure adjacent to the hippocampus. The amygdala evaluates the emotional significance of experiences and modulates how strongly they are encoded. This is why traumatic events, such as accidents or threats, are often remembered with remarkable clarity. The amygdala signals to the hippocampus that these events are important, releasing hormones like adrenaline that strengthen memory formation. Positive emotional experiences are also enhanced in this way, reinforcing connections between the amygdala and hippocampus. For skills and habits, known as procedural memories, other brain regions take the lead. The cerebellum and basal ganglia are crucial for motor learning and coordination. These structures allow you to ride a bike, play an instrument, or type on a keyboard without consciously recalling each movement. Procedural memory is implicit. You perform actions effortlessly once learned, even if you cannot articulate the exact steps involved. Memory retrieval, the act of recalling stored information, relies on a dynamic interplay among these regions. The hippocampus acts as a pointer, helping the cortex reconstruct the details of an experience. Emotional tone from the amygdala adds vividness and significance, while procedural memories may be triggered automatically by context or repetition. This networked approach is why memories are rarely perfect replicas of events. They are reconstructions influenced by multiple brain regions, context, and prior knowledge. The distributed nature of memory also explains why brain damage affects memory differently depending on the region. Damage to the hippocampus may prevent the formation of new memories but leave older ones intact. Cortical damage can erase specific knowledge, while amygdala injury can flatten emotional memory. The brain's redundancy and interconnectedness, however, allow it to compensate and adapt in remarkable ways. Memory is a collaborative effort of specialized brain structures. The hippocampus organizes new experiences, the cortex stores knowledge, the amygdala imbues emotion, and the cerebellum and basal ganglia manage skills. Together, these regions create the rich tapestry of human memory, allowing us to learn, adapt, and navigate the world. Understanding where memories live illuminates not only how we remember, but also why memories can be so vivid, so selective, and sometimes so fleeting. Forming a memory is only the beginning of the brain's remarkable process. Encoding an experience transforms it into a neural pattern, but that pattern is fragile and can easily fade if not strengthened. This is where memory consolidation comes into play, the process by which short-term memories are stabilized and transformed into long-term memories. Consolidation ensures that important experiences are preserved, integrated, and retrievable, while trivial information can be discarded to maintain mental efficiency. Memory consolidation occurs on two levels: synaptic consolidation and systems consolidation. Synaptic consolidation happens within hours of learning. It involves biochemical changes at the synapse, where neurons communicate. These changes strengthen the connections between neurons, a process essential for making the memory more durable. For example, if you meet someone new and learn their name, synaptic consolidation helps your brain solidify the neural pathways associated with that name so it sticks in your mind. Systems consolidation, on the other hand, occurs over longer periods, weeks, months, or even years. During this process, memories initially stored in the hippocampus are gradually reorganized and integrated into the cortex. This transition allows the memory to become more stable and less reliant on the hippocampus. That is why you might recall an event from your childhood years later, even if the details have shifted slightly. The core memory has been transferred and woven into your broader network of knowledge. Sleep plays a crucial role in memory consolidation, acting as the brain's internal organizer and processor. During slow-wave sleep, SWS, also known as deep sleep, the hippocampus replays recent experiences in a compressed and repeated manner. This replay strengthens the synaptic connections formed during learning, helping transfer memories to long-term storage in the cortex. In essence, deep sleep acts like a nightly rehearsal for your brain, ensuring that important information is cemented while trivial details fade away. REM sleep, the stage associated with vivid dreaming, further enhances memory by integrating emotional content and connecting new information with existing knowledge. During REM sleep, the brain not only strengthens memory traces but also reorganizes them in ways that improve problem-solving, creativity, and emotional understanding. This is why a good night's sleep often helps you approach challenges with fresh insights or remember something you struggled to learn the day before. the quality of sleep dramatically affects memory retention. Poor sleep or irregular sleep patterns can impair both synaptic and systems consolidation, leaving you more prone to forgetting. Studies have shown that even short-term sleep deprivation can reduce hippocampal activity, making it harder to encode new memories and recall existing ones. Conversely, consistent, restorative sleep enhances learning, strengthens memory, and improves overall cognitive performance. Beyond sleep, repetition and emotional engagement also enhance consolidation. Memories that are rehearsed, revisited, or tied to meaningful experiences are more likely to survive the brain's natural pruning process. Conversely, memories that are ignored or deemed unimportant may weaken and eventually fade. In this way, consolidation is selective, allowing the brain to optimize memory storage for relevance and significance. Memory consolidation transforms fragile, short-term experiences into stable, long-term knowledge. Synaptic and systems consolidation work in tandem, while sleep, especially slow-wave and REM stages, acts as a natural amplifier, strengthening and integrating memories. Repetition and emotional significance further ensure that what matters most is retained, while unimportant details are discarded. Understanding this process reveals why sleep, focus, and meaningful engagement are not just beneficial but essential for memory, learning, and cognitive health. While we often view forgetting as a flaw, it is actually one of the brain's most vital functions. Memory is not simply about storing every experience forever. It is about prioritizing, filtering, and maintaining information that is relevant. Forgetting allows the brain to avoid overload, maintain efficiency, and focus on what truly matters. In this sense, forgetting is not a failure. It is a sophisticated cleanup mechanism that keeps our minds functional and adaptive. One of the primary ways the brain forgets is through synaptic pruning. Just as neurons strengthen certain connections to solidify important memories, unused or weak synapses are eliminated over time. This pruning process allows the brain to optimize its network, improving efficiency and making it easier to learn new information. Imagine your brain as a garden. Strong, healthy connections flourish, while weeds and dead branches are removed to make room for growth. Without pruning, the brain would quickly become cluttered, slowing cognitive performance and impairing learning. Another mechanism of forgetting is memory decay, the natural fading of memories over time. Memories that are rarely recalled or rehearsed tend to weaken gradually. This applies to both short-term and long-term memories. For example, you might remember the name of a classmate from years ago if you frequently think about them, but forget the names of dozens of people you met briefly once. Decay is a normal part of memory's life cycle, allowing the brain to discard irrelevant or obsolete information. Interference is another common factor in forgetting. Memories can compete with one another, and sometimes new information disrupts older memories or similar memories overlap, making recall difficult. For instance, learning a new phone number may temporarily interfere with your recall of an old one. Interference demonstrates that memory is not a static repository but a dynamic network in which retrieval depends on context and competition between stored information. Interestingly, the brain also employs motivated forgetting, actively suppressing memories that are emotionally painful or traumatic. This defense mechanism can help protect mental health by reducing stress, anxiety, or trauma-related distress. Psychologists refer to this as repression, where certain experiences are rendered less accessible to conscious awareness. While repression can shield the mind from immediate pain, it can sometimes make it harder to recall and process these events later. Forgetting is not merely random, it is selective and purposeful. The brain prioritizes information that is relevant, meaningful, or frequently used while discarding details that are less significant. Emotional and personal relevance heavily influence what is retained. Positive or negative experiences that carry strong emotional weight are more likely to persist, while mundane events are more likely to be forgotten. This selectivity ensures that memory remains efficient, adaptive, and aligned with survival and learning. At the molecular level, forgetting involves processes like long-term depression, LTD, which weakens synaptic connections. Just as long-term potentiation, LTP, strengthens connections to solidify memories, LTD reduces synaptic strength, allowing the brain to erase unnecessary information. This delicate balance between remembering and forgetting is essential for cognitive flexibility, allowing us to learn, adapt, and respond to new challenges. Forgetting is an essential part of the brain's memory system. Through synaptic pruning, decay, interference, and motivated forgetting, the brain maintains a dynamic balance, preserving what is meaningful while eliminating what is unnecessary. Forgetting is not a weakness. It is a sophisticated, adaptive mechanism that ensures memory serves our learning, decision-making, and survival. Understanding this cleanup process reveals that memory is not just about accumulation, but about efficient, purposeful management of information, highlighting the brain's incredible ability to prioritize and adapt. Memory is not just a mental phenomenon, it is a molecular one. At its core, remembering and forgetting are processes governed by the intricate interplay of neurons, synapses, neurotransmitters, and proteins. Understanding the molecular mechanics behind memory reveals how the brain translates fleeting experiences into lasting knowledge and, conversely, how it erases information that is no longer needed. The foundation of memory lies in the synapse, the tiny junction where neurons communicate. When one neuron sends an electrical signal to another, it releases chemical messengers called neurotransmitters, which bind to receptors on the receiving neuron. This communication creates a chain reaction of electrical and chemical activity, representing information in a form the brain can store. One of the most critical mechanisms for memory formation is long-term potentiation, LTP. LTP strengthens the connections between neurons, making it easier for them to fire together in the future. Repeated activation of a neural pathway reinforces the synaptic connection, essentially writing a memory into the brain. For example, if you repeatedly practice a skill or recall a fact, the corresponding neural circuits become more efficient, which is why practice improves retention. LTP is considered a cornerstone of learning and long-term memory formation. Neurotransmitters play a pivotal role in determining which memories are encoded and how strongly. Glutamate, the brain's primary excitatory neurotransmitter, is essential for synaptic plasticity and LTP. Dopamine, often associated with reward, signals the importance of an experience, making it more likely to be remembered. Acetylcholine is another key player, involved in attention, learning, and memory consolidation. The precise orchestration of these chemicals ensures that memories are encoded efficiently and meaningfully. Beyond neurotransmitters, protein synthesis and gene expression are crucial for stabilizing memories. After a memory is initially encoded, neurons must produce specific proteins that strengthen synaptic connections. This involves activating certain genes to generate structural proteins, enzymes, and receptors that reinforce the memory trace. Without this molecular support, memories remain fragile and prone to decay. Just as memories are written at the molecular level, they can also be erased or weakened. The brain uses processes like long-term depression, LTD, to reduce the strength of synaptic connections. LTD selectively weakens neural pathways that are less frequently used, effectively erasing memories that are no longer relevant. This is how old habits can be unlearned and why certain details naturally fade over time. Memory erasure is not purely passive, it can also be actively regulated. The brain may suppress traumatic or stressful memories to protect mental health, a mechanism supported by molecular changes that reduce synaptic activity in circuits associated with those memories. Similarly, forgetting irrelevant information prevents cognitive overload, allowing the brain to prioritize learning and adaptation. In essence, memory is a dynamic molecular dance. Electrical signals, chemical messengers, gene expression, and protein synthesis work in harmony to write, stabilize, and sometimes erase the information that defines who we are. Every thought, skill, and experience leaves a molecular imprint on your brain, and the delicate balance between potentiation and depression determines which memories endure and which fade. By understanding these molecular mechanics, we gain insight into the brain's incredible ability to learn, adapt, and evolve. Memory is not merely a collection of past experiences. It is a living, constantly reshaping network that reflects the intricate molecular symphony playing out in billions of neurons every second of your life. Memory is not a purely mechanical process. It is deeply intertwined with emotion and personal meaning, which shape how experiences are encoded, stored, and recalled. The brain does not treat all memories equally. Some fade quickly, while others remain vivid for decades. Emotion and meaning act as a natural amplifier, determining which moments are etched permanently into our neural networks. The amygdala, a small almond-shaped structure in the brain, plays a central role in this process. It evaluates the emotional significance of an experience and interacts closely with the hippocampus, the brain's primary memory-forming hub. When an event triggers a strong emotional response, whether fear, joy, anger, or surprise, the amygdala releases neurotransmitters like adrenaline and cortisol. These chemicals heighten the brain's alertness and enhance synaptic connections, making it more likely that the memory will be encoded deeply. This explains why emotionally charged events, such as a first love, a major achievement, or a traumatic accident, are remembered with striking clarity. Emotion also affects memory retrieval. Memories tied to strong feelings are easier to access and often more detailed. In contrast, neutral or mundane events may be difficult to recall because they lack the emotional anchor that guides attention and strengthens neural connections. The emotional intensity of a memory creates a kind of imprint, signaling to the brain that this information is important and worth retaining. Beyond raw emotion, personal meaning plays a crucial role in memory retention. Experiences that connect to one's identity, values, or life goals are more likely to be remembered. For instance, a lesson that directly relates to your career, a pivotal decision in a relationship, or a defining life challenge will leave a stronger impression than an unrelated fact or trivial observation. The brain prioritizes information that is relevant to survival, growth, or social belonging, ensuring that memories serve both functional and psychological purposes. Interestingly, emotion and meaning can also distort memory. Highly emotional events may be remembered vividly but not always accurately. The brain tends to reconstruct memories during recall, often influenced by beliefs, expectations, and context. This is why two people can recall the same event differently, or why details of a past experience can shift over time, even if the core emotional tone remains intact. Emotion shapes memory not just in what we remember, but in how we remember it. Emotion also influences how memories are integrated. During REM sleep, emotional experiences are often replayed and linked with existing knowledge and memories, creating patterns of understanding and insight. This process helps us make sense of experiences, extract meaning, and sometimes even generate creativity. A vivid dream, for instance, may not be an exact replay of reality, but it can reinforce emotionally significant memories and integrate them into our broader life narrative. Emotion and meaning act as the brain's natural filters, determining which experiences are preserved and which fade. They ensure that memory is not just a catalog of events, but a living archive of what truly matters. By amplifying relevant experiences, providing emotional context, and connecting new information to existing knowledge, these forces transform simple neural patterns into memories that guide behavior, shape identity, and define human experience. Understanding this relationship reveals a profound truth. Memory is not merely a storage system, but a reflection of our values, priorities, and emotional landscape. Every memory we retain, every detail we forget, is shaped not just by neurons and molecules, but by what we care about, what moves us, and what gives our lives meaning. Memory has always fascinated scientists, philosophers, and educators, and modern neuroscience has opened doors to understanding, enhancing, and even manipulating recall in ways that were once the realm of science fiction. Today, research is revealing how we can improve memory, intervene in the retention of traumatic experiences, and influence how memories are consolidated and retrieved. One of the most well-studied approaches to enhancing memory is through cognitive techniques. Methods such as spaced repetition, mnemonics, and chunking leverage the brain's natural encoding and consolidation mechanisms. Spaced repetition, for example, involves reviewing information at increasingly longer intervals, which strengthens synaptic connections and reinforces long-term potentiation, LTP. Mnemonics create meaningful associations, making information easier to encode, while chunking organizes complex data into manageable chunks, aiding both short-term and long-term recall. These strategies exploit the brain's inherent plasticity, allowing us to maximize retention efficiently. Sleep is another critical factor in memory enhancement. As discussed in earlier sections, both slow-wave sleep and REM sleep play pivotal roles in consolidating memories. Studies have shown that optimizing sleep patterns, ensuring sufficient duration, consistency, and quality, dramatically improves memory retention, problem-solving abilities, and creativity. Napping strategically after learning sessions can also facilitate consolidation, giving the hippocampus and cortex time to process and integrate new information. Modern neuroscience has also begun exploring pharmacological approaches to memory enhancement. Certain drugs and supplements influence neurotransmitters, synaptic plasticity, and neurogenesis, potentially improving memory performance. For instance, compounds that increase acetylcholine activity can enhance learning and recall, while other interventions may target dopamine pathways to strengthen reward-based memory formation. However, these approaches remain experimental, and the ethical and long-term implications of chemically altering memory are still under intense scrutiny. Beyond enhancement, scientists are investigating ways to manipulate or suppress specific memories, with profound therapeutic potential. Techniques such as propranolol administration shortly after a traumatic event can dampen the emotional intensity of the memory, reducing symptoms of PTSD. Similarly, optogenetics, a cutting-edge method used primarily in animal studies, allows researchers to activate or suppress specific neural circuits associated with certain memories. While these techniques are still largely experimental, they suggest the possibility of selectively modifying memory in humans, potentially alleviating trauma or maladaptive behavioral patterns. Brain stimulation technologies are another frontier in memory research. Transcranial magnetic stimulation, TMS, and transcranial direct current stimulation, TDCS, can non-invasively enhance activity in targeted brain regions, improving memory recall and learning efficiency. Early studies indicate that these methods can enhance working memory, facilitate long-term learning, and even modulate emotional aspects of memory. These interventions highlight the incredible malleability of the human brain, revealing memory as a dynamic process that can be shaped by both biological and environmental factors. Importantly, these advances raise profound ethical and philosophical questions. Memory defines identity, shapes personal narrative, and influences behavior. The ability to enhance, erase, or manipulate memories challenges our understanding of self, responsibility, and human experience. While the potential to improve learning or reduce trauma is extraordinary, it also underscores the need for careful ethical oversight. Modern science is illuminating both the power and flexibility of memory. Cognitive strategies, sleep optimization, pharmacology, and advanced brain technologies all demonstrate that memory is not fixed. It is dynamic, malleable, and profoundly influenced by internal and external factors. As we continue to explore these frontiers, we gain not only practical tools to enhance learning and well-being, but also deeper insight into what makes us human. Our memories, our experiences, and the intricate neural dance that preserves them. Memory is one of the brain's most extraordinary capabilities. It allows us to learn from the past, shape our identity, navigate the present, and imagine the future. Through memory, we preserve knowledge, skills, and experiences, creating a rich tapestry of who we are. Memories connect us to loved ones, guide our decisions, and inspire creativity. They are the foundation of human consciousness, enabling us to understand ourselves and the world around us. Yet, memory is not infallible. It is remarkably fragile, subject to distortion, decay, and erasure. Memories can fade over time, blend with other experiences, or be reshaped by emotion and context. The same neural systems that preserve memories also allow them to be altered or lost, highlighting the delicate balance between retention and forgetting. Even vivid recollections are often reconstructions rather than perfect recordings of reality, shaped by perception, expectation, and meaning. This dual nature, powerful yet fragile, is what makes memory both fascinating and essential. Forgetting is as important as remembering, allowing the brain to prioritize, adapt, and maintain efficiency. Emotion and significance determine which experiences are etched permanently and which are discarded, ensuring that memory serves both survival and personal growth. Modern science continues to uncover the molecular, structural, and functional mechanisms underlying memory, revealing its dynamic and malleable nature. Ultimately, memory is a living archive, constantly evolving as we experience, learn, and reflect. It is a reminder that our minds are not static storage devices but active, adaptive systems. By understanding the power and fragility of memory, we gain insight into what makes us human. Our thoughts, our emotions, our experiences, and the delicate threads that weave the story of our lives.